Hi folks, we're going to start into chapter 4 this time. Very interesting, to me at least, genetics and cellular function. We're going to be talking about DNA, RNA, cellular transportation, lots and lots of different things. So I don't want you being overwhelmed. This is a quite involved chapter, but if you just try your best, try to learn the basics again, you'll do okay. I'm going to record this in 2 different sessions because I tend to get wordy, and sometimes it's too long to put into YouTube. I'm also sending you through a message attachment a list of the figures and tables in chapter 4 that I hope that you could print and have available as we talk. I remember when I learned this chapter the very first time, or this material, a long time ago, it helped me to look at pictures along with the words. Usually, I learn best just by reading. But for this, when we talk about chromosomes, DNA, RNA, how proteins are made, a picture along with the words makes things much, much easier and easier to understand. So if you can, print out the tables and the figures that I have sent, or I will send with this, it'll make things a lot easier. Okay, we're starting today. We're going to do sections 4.1 and 4.2. Now, first, again, please go to the study guide on page 137. And if you don't print off anything else, print that, please. Section 4.1 talks about DNA, RNA, and the nucleic acids. 4.2 talks about genes and how they work, what they are. So this part of the chapter will be longer than the second half. Now, DNA and RNA are the 2 types of what are called nucleic acids that exist in our body. Now, I want to just read something from the text here. With improvements in the microscope, biologists of the late 19th century, that's the 1800s, saw that cell division is immediately preceded by nuclear division. And during nuclear division, the chromosomes split in 2 and distribute their halves to the 2 daughter cells. Okay. A famous biochemist that did a lot of work in this field, Johann Friedrich Miescher, 1844 to 95. He did a lot of work with a microscope and studied the nuclei of white blood cells. In 1869, he discovered an acidic phosphorus-rich substance that he named nuclein. He correctly believed that this to be the cell's hereditary matter, although he was never able to convince other scientists of this. We now call this substance DNA, deoxyribonucleic acid, and we know that it contains all of our genetic material. Now, please go to figures 4.1 and 4.2. All of the DNA all of the genes and chromosomes of our body are made up of only 5 different nucleotides. Look at part A of figure 4.1. That is a nucleotide. It has a sugar, here in this case deoxyribose. It has a phosphate group, and then an adenine. These are either purines or pyrimidines. There's adenine, guanine, cytosine, thymine, and uracil. These are the 5 bases that are pictured in figure 4.1, part B. This is truly amazing. Only 5. And to make it even more amazing, because of the chemistry of these, there are only certain pairs that will bond together. If you look at figure 4.2, look at the bonds, the hydrogen bonds that exist. Adenine will only bond to thymine and vice versa. Guanine will only bond to cytosine and vice versa. Now, uracil we'll talk about later because it's only present in RNA. Okay, just look at that DNA. That truly, truly amazes me. James Watson and Francis Crick are the gentlemen from England who came up with this model showing that DNA exists with 2 strands in a double helix form. It's amazing. It's beautiful. Okay, now some more definition. DNA does not exist just as a naked helix all by itself floating around. It is complexed with proteins to form a fine filamentous material called chromatin. And by the way, I kind of skipped over this, DNA is only found in the nucleus of the cell. Now, the chromatin, in most cells, this chromatin occurs as 46 long filaments, and these are called chromosomes. We have 23 pairs of chromosomes that we'll look at in just a bit. Figure 4.4 shows the chromatin structure. This is a phenomenally good radioscope picture. On the left, part A, you can see the 50 nanometers mark. There are 100, got this written down somewhere, 100 billion nanometers in 1 meter. So this is pretty tiny. And this was totally unknown until we came up with the phenomenally good microscopes that are available. Now, look down through 4.4 again. You will see the DNA helix that's wound around a core. And then finally at the bottom you see what is called the chromosome. That is, these have many, many, many different pieces of, well, no, they're all one piece of DNA, but innumerable different base products there. Okay, table 4.1 shows a comparison between DNA and RNA. And this is something that you should know the basics of. DNA, the sugar in DNA is deoxyribose. The sugar in RNA is just ribose. The nitrogenous bases in DNA, as I mentioned, are A, T, C, and G. They'll stand for the 4 basic bases. In RNA, uracil, U, replaces the thymine. You'll see also by the number of bases that DNA is huge compared, I have to get my glasses on here, compared to DNA or RNA. DNA has 10 to the 8th. 10 to the 8th base pairs. That's 100 million. Where RNA only has 70,000 to 10,000. Okay, I'll leave the rest of that for you to look at. Now, let's talk a little bit about RNA. Ribonucleic acid. These are smaller, and these can exist either in the nucleus or out in the cytosol of the cell. There are 4 different kinds of RNA. Messenger RNA with a little M in front, ribosomal RNA with an R, and transfer RNA with a T. We will talk about these more when we start talking about protein synthesis. As we've talked about, RNA is much smaller, and it is able actually to go out through the nuclear pores and can go back and forth from the nucleus into the cytosol and back and forth. Now, what is a gene? And I don't mean your blue jeans. The name gene, G-E-N-E, it's very simple. A gene is just a small piece of DNA that codes for the production of a specific molecule of RNA, which then goes on to code for the synthesis of one or more very specific proteins. So, what looks very, very complicated is actually pretty simple. Each gene basically ends up allowing the synthesis or directing the synthesis of one or more proteins. As I mentioned, the 46 human chromosomes, there are a different number of chromosomes in different creatures, exist in 2 sets of 23, and they are all paired. 22 of the pairs are identical. The 23rd pair are the sex chromosomes. In females are 2 X's, and in male it's X and Y. Now, another interesting thing, you may have heard of the Human Genome Project. This was done by numerous scientists around the world over about a 13-year period. And because of that project, we now know the base sequence, and therefore the complete structure of more than 99% of the chromosomes in our body. Okay, now just a brief mention about what is called the genetic code. The code, just like Morse code or any other code, is a system that enables the 4 nucleotides to code for the amino acid sequences of all proteins. Remember that proteins are made up of many, many different amino acids, of which there are 20. Now, the minimum code to be able to symbolize 20 different amino acids is 3. And that's the case in DNA. So, if you look over at table 4.2, you will see some examples of a base triplet. Let's look at the first row. CCT. That means somewhere on the DNA is cytosine, cytosine, thymine. And because of the complementary pairing, the codon of the messenger RNA that we'll talk about here shortly is GGA. Remember, cytosine only bonds with guanine. Thymine only bonds with adenine. There are several other codons, these triplets, that are very, very important. There are 3 which are called stop codons, and you can read about them there. One is called the start codon. It's the codon AUG in RNA. This codes for methionine, which is one of the amino acids. When the cell sees this, it knows to start making protein. Now we'll talk about protein synthesis. This now gets complex even more a bit, but I hope that you can print out and look at figure 4.6 on page 120, 4.7 on 121, and 4.8 on page 122. If you can look at these figures while you're reading or listening to me and do it over and over and over, suddenly the light bulb will come on. You say, oh, I see that. Now, I'm going to read a little bit more out of the book here. When a gene is activated, remember that the AUG is the activating codon. A messenger RNA is made. It's a mirror image of the portion of the DNA, the gene. And most of this messenger RNA migrates from the nucleus to the cytoplasm, where it serves as a code ultimately for the assembling of amino acids. The step from DNA to messenger RNA, you can see in figure 4.6, is called transcription, to transcribe something. And in genetics, that means it's the process of copying the genetic instructions from DNA, a portion of the DNA, to RNA. And this requires an enzyme called RNA polymerase. I'm sorry, polymerase. Polymerase. One or the other. Okay. Now, the step messenger RNA you see in the third part down, after these have been split apart, this is called splicing, and then these triplets end up forming or being translated into an amino acid chain. So the messenger RNA carries the genetic code from the nucleus to the cytoplasm. The transfer RNA, oops, I don't want to skip over the messenger RNA through transcription and then translation ends up forming what is called a transfer RNA. And there's a picture of a typical transfer RNA molecule in figure 4.7. You see the hydrogen bonds between the parts of the cell, and you also see the codon at the very end. In this case, ACC. Now, ACC is not listed on the other table. Oh, there it is. It codes for the amino acid glycine. So this transfer RNA moves about the cytoplasm, locates and then binds this one specific amino acid called, forgot already. What did I say? What did I say? What did I say? I'm sorry. Oh, ACC. I don't remember all this, folks, so you can't remember it all either. Theanine. Okay. Now, so you can imagine this transfer RNA with the codon binding thymine and moving around in the cytoplasm. And then finally, it is picked up by a ribosome. We talked about ribosomes back in chapter 3. Your author calls them little reading machines. They're found in the cytosol, freely floating, or on the outside of the rough endoplasmic reticulum, or on the outside of the nuclear envelope. You may want to go back to chapter 3 and just take a look at that again. Now, each ribosome has two parts, a small subunit and a large subunit. These are floating around separately and only come together when the translation of the messenger RNA is actually happening. Now, the translation. Look now, I told you about figure 4.8. And if you go from 1 to 2 to 3 to 4. Now, let's look at part 1 just to see where we are. In the left lower corner is this outer cell or outer membrane of the nucleus of the cell. And there you can see messenger RNA not escaping, but they're coming out into the cytosol through the nuclear pores. And then they go looking for a ribosome. And you can see the ribosomes are shown in purple. And these are located in several different locations, as I mentioned. And they pair up. The large subunit and the smaller subunit pair up on the messenger RNA, and then imagine that picture being made much larger in part 2. Each transfer RNA is carrying around one amino acid, and when it comes in contact with the ribosome, this one amino acid is released and ends up forming a chain of amino acids. And then the transfer RNA is released and goes out looking for another amino acid. Part 3 just follows this sequence around, and then you see a free protein finally. This is just a long chain of amino acids. That's all it is. Part 4 shows, again, a ribosome releasing this chain of amino acids into an endoplasmic reticulum. And these are now then they're packaged in sometimes into transport vesicles that can move around the cytoplasm or actually out of the cell. I mentioned then that there are 3 different termination codons: UAG, UGA, and UAA. Notice the U for uracil, so these are only found on the RNA. When one of these stop codons comes into play, the whole process is terminated, and we have, hopefully, a correctly synthesized protein molecule. Now again, look at oh, I'm blanking on the name of the microscope. I wish I had somebody was here in class with me. Electron, there it is, electron microscope. Okay. There you see again the 60 nanometers. You could not possibly see this anywhere except with this electron microscope. You can actually see the ribosomes. You can actually see the chains of amino acids, the messenger RNA. This to me is phenomenal, just phenomenal. Okay, now there's another 4.10 is just another picture to show you. It starts with the DNA double helix and ends up with 6 different amino acids being linked together with what are called protein bonds to form a very short piece of protein. Now, what happens to this protein? Picture or figure 4.1 shows some of the different things that can occur. Some of it is released into the cytoplasm. Some is packaged into small transport vesicles that bud off of the endoplasmic reticulum. And some go to the Golgi complex and form into Golgi vesicles that are actually these then migrate to the outside of the cell, and these proteins are released to the outside of the cell. Okay, with that, I am going to stop. We've covered relatively well, I think. I'm humble too. Sections 4.1 and 4.2. You will, I'm going to go into now 4.3 and 4.4. Thank you for your attention.